All right, let's start with a sound check. Can you hear me in the back row there? Can you hear me in the back row there? Excellent. Just want to start with, are there any remaining questions about the exam or about what we talked about enzyme kinetics on Monday? Any common questions? One that I've gotten a couple of times is, are the essay grades up yet? And no, they're not. Essay scoring is a human fine art and is done by hand and eye, and it doesn't happen nearly as fast as machine grading. We will announce when those grades are up. You don't need to ask. We will announce it, okay? At some point, they will be up. Any other questions then about the test? Okay. Let's think a bit about enzyme kinetics then. Enzyme kinetics sometimes has this kind of horror twitch reaction that people have about it. It just seems to evoke fear and loathing in some people, but it shouldn't give you that sensation. It's really a tool, and it really tells us useful things about what enzymes can and can't do. So I want to review a couple of things from Monday, and then we're going to go on through the inhibitors and see how that sort of illuminates that further. Just have to get my pointer options up here. There we go. So you remember we were talking about there being some there's a Vmax, the fastest possible turnover that an enzyme can do when completely saturated. So it's a measure of how quickly it does the catalytic activity when it's occupied. So it's measuring, if you like, what's happening at the catalytic residues in the active site. You have another parameter which measures, in many cases, approximately affinity, the tendency of the enzyme to bind a substrate. And because the Vmax is approached through a hyperbolic curve, you can't just simply use what it takes to get to Vmax as a number. We need a more flexible number than that. And so the strategy of looking at the half maximal value, which is the same we used for pKa, it's the same we used for oxygen binding. If we look at 1/2 of the maximal volume of velocity, so down half, and we look at the curve then, the concentration of substrate that it takes to get half the maximal velocity is called the Michaelis constant. It's, roughly speaking, a measure of affinity, the tendency for a substrate to bind to the enzyme. Vmax and Km are completely independent of each other. There is no necessary logical connection between them. You can have a rapid Vmax and a low affinity. You can have a high affinity and a low Vmax, and any combination thereof. There is no necessary logical connection between Vmax and Km. The two are independent properties of an enzyme. Are people comfortable with that? Do you get what I'm getting at, or do you want me to go through that one more time? Because 25 years, I've heard all possible combinations of things that aren't true about that, which I'm not going to repeat because then I'll confuse you. Okay. Well, then let's look a little bit further on. I wanted to look up. Now, because Vmax and Km are independent variables, you could have a case where something is a very rapid enzyme when occupied, but it's not very good at binding substrate. It has a weak affinity or a large Km. You could have something that binds it very well, but doesn't do the reaction very well when it's occupied. You would have a high affinity, a small Km, but a low Vmax. So to really compare one enzyme to another, you want a number that captures both of those. And that's what the catalytic efficiency does, is it combines into one ratio, the Kcat, which is going to be proportional to the Vmax. It's a measure of the turnover. And the Km, which is, in general terms, approximately a measure for affinity. And so this ratio then allows you to compare amongst enzymes with one number that combines ability to bind the substrate, ability to catalyze the reaction. So that's refresher. We did that before. let's just refresh this one more time too. Using a hyperbolic curve is at best an approximate method, although now we have computer simulations that allow for better interpolation and extrapolation and so forth. But a powerful method that was used for a long time is a way of turning a curved line into a straight one. And I'm not going to repeat the derivation, but when you do that, when you compare the reciprocal of velocity to the reciprocal of the substrate, now enzyme activity becomes a straight line, and you can read off of it the intercept on the velocity axis, the reciprocal of the Vmax, and you can read off of the substrate axis the reciprocal, the negative reciprocal of the Km. Remember that as you go up on a graph like this, you're going to smaller numbers because this is a double reciprocal. So this linear representation allows you to capture those variables more exactly than trying to do it through a curved line. Remember that the vertical axis is always going to involve something about velocity, either directly or reciprocally. The horizontal axis is the substrate concentration that's going to be the independent variable. Okay. So, sigmoid, yeah. And then, just refreshing you again, when you see sigmoid curves of almost any kind in biology, in biochemistry, something cooperative is going on. And you can have enzyme activity that is sigmoidal in its behavior. So as again, substrate concentration starts to rise, not much change in velocity, then a linear increase in velocity or quasi-linear, and then it tapers off. Again, the sigmoid shape, characteristic of cooperative behaviors. Can't linearize this with a double reciprocal plot. Double reciprocal won't work on a sigmoidal curve. But the Vmax meaning is still the same meaning. An enzyme with sigmoidal kinetics can still have a Vmax. That's how fast it'll go when it's saturated. It'll still have a Km, a concentration of substrate that will half saturate it, or how well it binds the substrate. Okay? So now we're going to go on with the enzyme inhibition that we got started on. We talked about suicide substrates that look like substrates and then become covalently attached. Then we started to talk about competitive inhibition. And here's the Pac-Man enzyme. And the schematic here is trying to indicate that an active site has an actual shape for which a substrate can fit. It's going to bind to the side chains on the folded protein and so on. But something else might also fit, like an inhibitor, but not be a substrate. Doesn't become covalently attached like the suicide substrates, simply can occupy that space. And in that case, we would see a curve like this. This is the sort of intuitive way of representing it. You can see here that a competitive inhibitor has no effect on the Vmax. That if there's enough of it around, it will turn over. But it appears to change the Km because you need more substrate to get to the same velocity. So you can detect a competitive inhibitor because it apparently increases the Km. It apparently decreases the affinity. You need more substrate around to get to the same velocity. Any questions? We can represent that on a Lineweaver-Burke plot, which is what we call the double reciprocal plot. And again, the vertical axis is the reciprocal of the velocity. The horizontal axis is the reciprocal of the substrate. And you can see that the lines intersect on the vertical axis because there's been no change in the Km. But the intercepts on the x-axis now are different because in the presence of an inhibitor, it takes our substrate to occupy the site, and it has an apparently larger Km, which is closer to the zero here because this is a reciprocal plot. Okay? Now, if you think about it, there are many enzymes where the product can also bind the enzyme and undergo a reaction, and in fact it can undergo the reverse reaction. Many enzymes, though not all, are reversible. And what we call that when the product can inhibit it is product inhibition. It's really a form of competitive inhibition. Here is an example of a reaction. Glyceraldehyde 3-phosphate is a molecule you're going to see many times. It's in carbohydrate metabolism, it's in lipid metabolism, it's in amino acid metabolism, so it's a good friend. And there's an enzyme that can rearrange glyceraldehyde 3-phosphate into dihydroxyacetone phosphate. And this particular enzyme happens to be an extremely good enzyme. Its name is down there in the corner if you're curious, triose phosphate isomerase. So it rearranges triose phosphates. Close to kinetically perfect, an extremely good enzyme. And so if you start off with pure glyceraldehyde 3-phosphate and let the reaction run for a bit, you're going to accumulate dihydroxyacetone phosphate. And that can then bind on the enzyme and inhibit it. So the product can act as an inhibitor. That has a practical consequence. It means that when you're measuring these things, you have to measure the velocity early on before you've made much of that product, which can act as an inhibitor. It also has biological significance. If the product is accumulating for some other reason, it will tend to inhibit that enzyme from going in that direction, just simply by competitive inhibition, apart from various allosteric interactions. If we look at the intermediate here between glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, we have what we call an enediol. So here's the ene, the double bond, and there's an alcohol, and this is an enediolate because the alcohol here has lost its hydrogen. We looked at this intermediate when we were looking at acid-base catalysis. So this is the transition state. This is the thing that is at the peak of the hill between Da3P and dihydroxyacetone phosphate. And so if you can find a compound that looks a lot like the transition state, you can imagine that would be a particularly good competitive inhibitor. And here is phosphoglycohydroxamate, which I believe actually may be one of the herbicides that's used, but I'm prepared to be corrected on that. But you can see, I think, how this structure looks like this structure. You have a double bond here, alcohol sticking off at the same angle, negatively charged oxygen there. So this compound could really nicely fit into the pocket of the enzyme in the transition state. But it can't be turned into dihydroxyacetone phosphate, so it'll compete for the reaction. Sometimes they can be really remarkably good inhibitors. Here's a different reaction. Here we're looking at turning adenosine into inosine. They're both purine rings, both the fused two rings. And the only difference is going to be here, we're going to change this amine here to a carbonyl oxygen. So that's where all the business action is in that carbon. And here is the transition state intermediate. At this point, you have a tetrahedral carbon where you have a single bond to the oxygen and you still have a bond to your nitrogen. So you can kind of imagine it looking like that sort of a thing. The inosine can act as a product inhibitor, but this thingy, which is an analog of the actual transition state, has the carbon in that tetrahedral shape, and it is inhibitory with an inhibitory constant of 10 to the minus 13. So while the product can be mildly inhibitory in this case, the transition state is orders of magnitude, in fact, an order of magnitude better at binding to and inhibiting the enzyme in a reversible way. This is still a competitive inhibitor, but it sure doesn't take much of it to inhibit that reaction. Questions about that before we move on? Okay. There are many possible ways of interfering with an enzyme. There's another case called mixed inhibition. In the case of a mixed inhibition, you have an enzyme of some kind which can bind a substrate and go between an enzyme and an enzyme substrate complex. But in this case, the enzyme has another site somewhere else on the enzyme that can bind compounds. And in the case of a mixed inhibition, if that second site, not the active site, but a different site, is occupied with that inhibitor, it can end up modifying both the affinity and the catalytic activity of the enzyme. It neither binds it as well, nor does it catalyze the reaction as well. And what that means in kinetic terms is that the inhibitor can lower the Vmax, that its absolute ability to turn over when occupied is not as good, a smaller turnover number. And it can raise the Km. It takes more of the substrate to be half saturated. So it's not binding anywhere near the active site itself. It's binding somewhere else. And in doing that, it's somehow or other changing the conformation of that protein, both in its ability to even bind substrate and its ability to flex and turn and rip around and change the substrate into something else. That's why it's called a mixed inhibition. We do see a few examples of something called non-competitive. A non-competitive inhibitor changes only Vmax. Same affinity as in the absence of the inhibitor, but something about the way it binds to that enzyme makes it a worse catalyst. It has a lower Vmax, a smaller turnover number. I think you can see why non-competitive inhibition might not be terrifically common. That if you can bind somewhere else in a way that changes catalytic activity, you'd have to do it very delicately and not also change affinity. So, in which case you'd have a mixed inhibition. Now, we've been talking about this sort of as if it's all about putting in inhibitors that, we're doing to interfere with a reaction or to study it. But inhibition is an extremely important part of the regulation of metabolism. Metabolism can't do everything at once, or it'd be a big, booming, buzzing confusion, and you'd be wasting energy and wasting materials. So one of the important parts we'll be studying in metabolism is how do you regulate metabolism? How do you do the right thing at the right time? And inhibition turns out to be one of the tools that organisms use for this in many different cases. One very important form of it is allosteric inhibition. And so you can have, here we have a miniature version of a pathway. This is an enzyme, a substrate called fructose 6-phosphate. We're going to see this one again. So this is a 6-carbon sugar. The phosphate's up here on the 6-carbon waving like a flag. And the enzyme phosphofructokinase is going to take a phosphofructose, which is a fructose with a phosphate, and add a phosphate or act as a kinase. So the product of phosphofructokinase is fructose 1,6-bisphosphate. 5-membered ring. This carbon still has its phosphate, but now the carbon over here has a phosphate. So you have fructose 1,6-bisphosphate. Some more steps happen in, little black box, then a miracle happens kind of explanations. And down at the end of the pathway, we have phosphoenolpyruvate. And phosphoenolpyruvate will itself have many interesting life paths it can follow. In looking at the regulation of this pathway, one of the compounds that can then regulate this is for that product of the pathway, down quite a long way, can come back and bind to the enzyme and inhibit its activity. It's not really product inhibition per se, because phosphoenolpyruvate is not a product of this enzyme. It's further down the pathway. But the structure of phosphofructokinase is such that it has binding sites for phosphoenolpyruvate, and when those are occupied, that modifies the activity of the enzyme. So we call that feedback inhibition. And as you can imagine, feedback inhibition is going to happen many times. A very useful tool. Here's looking at that reaction in just a little more detail. Phosphofructokinase is a multimeric enzyme with more than one subunit. And it again has a tensed and a relaxed state. You may remember from hemoglobin there was a tense and a relaxed state. And if we look at the relative behaviors of those two things, we really see some sort of dramatic changes. So in the green or the left-hand curve here, this would be a curve of activity, the change in the velocity of phosphofructokinase with substrate, typical kind of hyperbolic curve. Phospholipyruvate around, we see two dramatic changes. One is a change in the Km. So you can see that the Km here has increased many, many-fold. So it has a much lower affinity for the substrate. It's also changed the shape of the curve. It's gone from hyperbolic to sigmoidal. And so it's retained the ability to eventually get up there, but it does so now in a sigmoidal way rather than a simple hyperbolic way. The state that has the phospholipyruvate bound is called the tense state. It's tense, it's hard for the substrate to get into it, if you like to think of it that way. It's hard to bind, therefore a larger Km. ADP, phospholipyruvate, it can also be thought of as a symbol or a signal that the cell has adequate energy status. It's a high energy compound, and we're going to see what else it gets used for. So it switches phosphofructokinase into a less active state because you don't need to burn your sugar. Maybe this would be a good time to take your sugar and put it away as glycogen or make fat out of it. ADP is the product of a lot of reactions that use ATP. So it is a metaphor or a symbol or a sign of a lower energy status in the cell. And ADP can help keep phosphofructokinase relaxed or more active. So one enzyme can have multiple different allosteric binding sites where many different compounds can feed information in. And some of these regulatory enzymes end up being small molecular computers where they can accept inputs from 5 or 6 different compounds and process them all and come out with a correct end state that's appropriate for the current metabolic condition of the cell. I talked about hemoglobin as a little molecular computer. Some of these enzymes take that to another degree. Okay. Where are we? Okay. So far we've talked principally about binding either at the active site or binding somewhere else on the enzyme, but there are in fact a number of other ways that enzyme activity can be changed as well. So for instance, you can change how quickly you synthesize or how quickly you degrade the enzyme. We tend to be aware of synthetic rates. In the last recent period, we've become more appreciative that degradation rates are every bit as significant as synthesis rates because it's the balance of the two that determine the steady state concentration. So anything that either increases synthesis or decreases degradation will overall raise levels. Anything that inhibits synthesis or raises degradation will lower levels overall. Some examples of how dramatic that can be, one of the key enzymes in gluconeogenesis is controlled by a hormone at the cell surface, and it can bind at that receptor, send a signal to the nucleus, have it transcribe, process the messenger RNA, and get it out into the cytoplasm to make new protein in a matter of 15 to 20 minutes. Which is actually faster than some of the largest cytoskeletal proteins can be translated. So changes in synthetic and degradation rates can be quite important. We can have changes in location. So for example, if you have something that's active only on the surface or something that's only active on the inside, by putting it in a different location, you take it away from wherever its substrate is and it can't be active. One of the reasons they're showing that particular one here as something going in and back from the membrane is we're going to see a number of membrane proteins where their activity is in part regulated by location. Some things are regulated through ionic signals, for instance, that the presence of calcium can bind to the enzyme in a way that turns it on or turns it off, or that binds to a small protein, which then binds to the enzyme and turns it on or turns it off. Some enzymes are sensitive to pH and go more or less active as the pH changes. There are some enzymes that are actually changed through covalent changes, where a group is actually covalently attached to the enzyme and stays on it until something leads to its being taken off. Two of the common forms for that would be phosphorylation, and we're going to see a number of examples of enzyme activity changing through phosphorylation. You think about how small a phosphate group is compared to how big an enzyme is, and it's really kind of amazing that a phosphate group can come close to turning on or turning off a whole enzyme. But we go back to the doorstop idea that, a small doorstop this big can keep the entire door open, and things like that can happen with phosphorylation. Lipids can also be used to modify enzymes as well as some other things. So I want you to talk to each other for a little bit about these questions. If you have a mutation at the catalytic site, which is more likely to change, the Km or the Vmax? And comparably, if you have a mutation at the specificity site or a specificity pocket, which is more likely to change, the Km or the Vmax? So chat among yourselves to see if you can come to a consensus, okay? This will be thrilling for people who are watching the video. They're going to be wondering what we're doing in the room. Anybody want to venture a comment on the first one? If you have made a mutation at the catalytic residues, which parameter do you think will be most dramatically affected? Sorry? Can someone raise a hand and shout at me? If we change the catalytic site, which one is most likely to be changed? Okay, would you like to take a stab at explaining your answer? It is, it is reasonable to think the probability will be with it. It's not a cast in iron thing, but the probability will be that if you change a mutation at the catalytic site, the catalytic residues that are actually involved with the making and breaking of bonds, the most likely thing to change will be the Vmax, its ability to turn over and make the reaction happen. Sometimes the catalytic residues can tolerate a change. So there are, for instance, families which have serines or threonines as the reactive residue, and you can think of serine and threonine and how similar they are, just a little difference in size, basically. But it can certainly be the case that a substitution at the catalytic residues is sufficiently small to leave the rest of the enzyme structure intact, so binding can happen, but take away key residues for the catalytic reaction itself. You remember in chymotrypsin you had the catalytic triad of the aspartate, the histidine, the serine, and you need all three of those guys in there. That's such an effective triad because, we know it's an effective triad because it got reinvented 5 different times. And so you can imagine if you were to change that aspartate to an asparagine, probably would not be a very good enzyme, but such a small change, probably no change in the binding. Well, having talked about that one, probably the next one is pretty obvious too. Which would be most likely to change if you mutate the specificity pocket, the Km or the Vmax? Okay, you want to take a stab at it? Okay, so in other words, a change of the specificity site may well alter the ease with which a substrate can fit into there and bind it all. But if if it still allows the scissile bond or the things that are actually going to be changed to come into contact with the catalytic residues at all, it probably can still have a measurable velocity, or maybe even the identical velocity Vmax once it's saturated. It might take 100 times as much substrate to get there. But once you saturate the enzyme, the catalysis might be intact. Now, do you think that mutations at either of these sites tend to have pure effects? Does it tend to be just Vmax that changes or just Km that changes? I'm seeing a lot of shaking heads. The enzyme is an entire structure. It's not made out of Legos or something, and perturbations in one place can propagate. So it isn't very common that you have a pure effect on one or the other. But if you have a clear notion of what catalytic residues are, then you can see why they are going to be what control Vmax. And if you have a clear notion of what a specificity pocket is, about how it's binding the rest of the substrate, you can see why that might have an effect on Km. So structure is related to kinetics. Enzyme mechanisms are related to kinetics. They're not separate chapters, they're all together. These things can have a very big importance in medical terms as well. And we could spend an entire semester actually discussing inhibitors that are medically important. But let's just talk about one in particular because it's a topic with such current concerns. HIV, of course, is the human immunodeficiency virus, really a terrific problem. To some extent, certainly in the United States still. In Africa, there are now millions of HIV orphans. So HIV and AIDS is one of the major medical challenges the world is facing right now. What is it that it's so terrible, and what can we do about it? Well, if we look at the structure of HIV, it is a single-stranded RNA virus. And so in the electron microscope, it looks kind of like this, has a core and a periphery. And that core, if we look at it more closely, is made up of RNA. So there's RNA molecules that are its genome. There are some enzymes packed in there, some proteins to do their work also, and then a coat that goes all around it of both lipids and proteins. So a dastardly efficient little machine. Before the enzyme can do anything very terrible, it makes more of itself. It's common for viruses to make one of their first gene products or first few gene products to be involved with making more virus. And so there's an enzyme called reverse transcriptase that has an activity that violates the central dogma as it was originally formulated. So what reverse transcriptase can do is take an RNA original and synthesize a strand of DNA that's complementary to it. So that's the first activity that the enzyme has at one of its active sites. There's a second active site that can degrade RNA. And so when that comes in, it will clean off the RNA molecule that was the template, leaving you with a single strand of DNA. And then that can become a template to become a molecule of double-stranded DNA. So single-stranded RNA has been turned into double-stranded DNA. The complementarity matches it, but you have to get rid of the RNA along the way. One of the major tools in fighting, or controlling, I should say, in controlling HIV and AIDS have been inhibitors of this key enzyme. Obviously, if the virus has a single-stranded RNA genome and it needs to turn it into DNA before it can make more viral proteins, that is a step you want to inhibit. And so let's take a little look at some of the inhibitors that have been used for that. So here's one of the first ones that was discovered called AZT for azetodideoxythymidine. The pharmacy world has a chemical name for everything, and then it has a generic name for them, and then they often have another name as well. So this is zidovudine, if you want to give it its generic name. But you can see how it's looking like a thymidine. It's got the thymine sticking up there. It's got the 5-membered ring, but it's got a dideoxy. It doesn't have an oxy there, so that would look like a DNA-like thing. But what's sticking off on this end will not allow another nucleotide to be added to it. So this is a chain terminator, just like the dideoxynucleotides were when we were sequencing DNA back several chapters ago. Here is another molecule that is a chain terminator. Cytosine here sticking up, deoxy here, deoxy there. So this is dideoxycytidine, and because it cannot be extended there, terminates the chain at that location. And you can see that these two things would terminate in slightly different places, but they would have fundamentally the same effect. There are other enzymes that can inhibit reverse transcriptase that are not analogs. So take a look here at nevirapine. This is not a chain terminator, but what does it look kind of like? Sort of. What do double-stranded nucleic acids have in the middle of them? Base pairs. So this kind of looks like the template or the footprint of a base pair. And probably one of the ways that it interferes is by intercalating and, gumming up the works that way. So one of the things that gets done in the case of HIV is to make one big protein called a polyprotein, and then to cut that up into pieces. And so here we have, this would be one polyprotein that's cut in 3 places to make 4 proteins. Here is another one that's a long one cut into 3 pieces, and so on. So these green ones are structural, they're part of building the enzyme. The purple ones are the enzymes, and then the blue ones are various kinds of accessory proteins that can regulate or stabilize or protect and so on. So obviously proteolysis is going to be important also for the virus to amplify and to be propagated in a way that will actually leave the cell. So proteases are key to the efficiency or the functioning of an HIV virus. And for that reason, the proteases became a second target for inhibiting the activity of the enzyme. And this is actually one of the early examples of something called rational drug design. Rational drug design is the idea that you know enough about the enzyme and the substrates to try from first principles to design something that will fit into that enzyme and inhibit it. And if we look at the normal place where the HIV protease cuts, this is a typical sequence for cutting here. So you can see here a phenylalanine side chain here and a proline next to it. So this is the sessile bond, the place where in the normal proteins it would be cut. And the rationale that they designed was to try to keep a bulky ring that could be where the phenylalanine is, and then make a mimic of peptideness hanging off the sides, and do little adjustments here and there on the molecule to make sure that it was soluble, that it was stable, and that kind of things. So here are a couple of examples of these protease inhibitors. And you can see here, there's the phenylalanine sort of sticking off, and that would be the sessile bond that's been inhibited. Here's the phenylalanine hanging off, that's the sessile bond. But these kind of look like peptides, don't they, hanging off to the side. So you can imagine how that would fit into the active site and how it could bind there but not be a target for cleavage. So it's acting as a competitive inhibitor. You would hope that because it's being targeted to the specific protease that HIV uses, it would be specific to HIV. They certainly tried hard, but these things do still bind at other sites and cause some side effects for people, despite their attempt to make it utterly specific for HIV. I'd like to walk you through a few of the figures then at the end of the book as a way to fix in your mind some of the concepts we've talked about. I won't do this for every chapter, but I think this is one where it's worth doing. So for this figure, which enzyme, you can talk among yourselves, which enzyme is going to have the higher Vmax? Which one has the higher affinity? Okay. So which one has the higher Vmax for the easy one first? B, okay. Which one has the higher affinity? And why does it have a higher affinity? Takes less substrate to get to that half maximal concentration. So this is one example of a pair of enzymes where one has a greater affinity but a smaller ability to catalyze the reaction. Which of these two would probably be allosteric? Right, because it's got this sigmoidal kind of character to its kinetics. This one's the hyperbolic, this one's sigmoidal, slow, linear, and then tapering off. If we look at this sort of family of enzymes, E1, 2, and 3, which of these enzymes has the highest Vmax? E3, okay, and that's based on?
It's the y-intercept.
Got it, exactly. So this is 1 over the velocity, and here it is the smallest on that axis, so the biggest number. So it has the greatest Vmax. Which of these 3 enzymes has the greatest affinity? Yes. Since this is a double reciprocal plot, a Lineweaver-Burke plot, this will be 1 over velocity, as we said before, the reciprocal of velocity. And this will be 1 over substrate, the reciprocal of substrate. Because enzyme activity won't look straight on a direct plot. It'll look either hyperbolic or sigmoidal. It's only on the double reciprocal plot that enzyme activities will be transformed into a linear appearance. Okay? Do you remember back when we were looking at the way of representing enzyme activity in a different way? Okay? So which of these then would have the greatest affinity? Yeah?
E2.
E2? And do you want to explain your answer?
Because it tracks closest to the.
Okay. But this is a reciprocal, remember. This is 1 over substrate. So this would be the larger. This will be the smallest. Okay, so the greatest affinity will be the smallest Km. This will be the smallest Km because it's the furthest from the axis. Double reciprocal plots are handy because they do transform things into straight lines. They're not as intuitive because you have to make allowance for the fact that they are double reciprocal plots. Let's take a little look at these guys. Acetylcholine is a transmitter, a neurotransmitter in the brain and in many other places. And decamethonium is an inhibitor that gets used to inhibit the activity of an enzyme called acetylcholine esterase. So here's acetylcholine. Here's the ester bond, right? So what kind of an inhibitor do you think decamethonium is? Competitive, mixed inhibitor, non-competitive? You can talk to each other. This is one time it's really not so much fun to watch the video. But they can work it out at home. If you're watching the video, pause it until you've worked out the answer for yourself. Does anybody want to take a stab at which part of the acetylcholine probably is interacting with the catalytic residues? Okay, so this is probably going to be interacting with the catalytic residues. And so what part of the enzyme is this part of the molecule probably interacting with? Some kind of a specificity pocket, something else that's there to hold it while the catalytic sites are doing it. So what part of the decamethonium molecule is probably going to be mimicking acetylcholine? So these are both tertiary amines that look like that end of it, and it's a symmetrical molecule, so either end could stick in. But will there be anything to interact with the catalytic residues? No, no. Whichever end sticks into the enzyme, there's nothing there at the place where you'd expect an ester, so it's probably acting as a pure competitive inhibitor, and probably a pretty good one since it's got two copies of the binding side, the specificity side. This is a very useful tool for neurobiologists, by the way, and I believe it's also used medically. Does anybody else? I think this is actually the drug that's used when you need to paralyze people on respirators. Not completely, just to keep them from fighting the respirator. What kind of an inhibitor is this inhibitor? Well, red is, the bottom line is with inhibitor. So the inhibitor that's shown, so this curve is in the absence of inhibitor, this is with the inhibitor. What kind of an inhibitor is this? Someone want to raise a hand so I can look in her direction? Or else shout loudly. You want to say it's a non-competitive? Okay. How do we know something is a non-competitive inhibitor? Okay. So what can we say about the Vmax in the presence of this inhibitor? What would you estimate would probably be the effect on the Vmax by this inhibitor? Not cooperative, did you say? If we look at the probable Vmax in the presence of this inhibitor, it probably has a lower Vmax. I mean, it's a little hard to guess from a hyperbolic, but that's a good guess. If we accept that this is a lower Vmax, what can we say about its Km? Well, its affinity is lower, its Km is higher, right? So in other words, if this is the new Vmax, it takes something like this much substrate to get it to half of the new lower inhibited Vmax. Whereas the uninhibited state, if this is the Vmax, it only takes this much to do it. So this inhibitor is affecting both the Km and the Vmax, the apparent Km, the apparent Vmax. So what kind of inhibitor does that make it? Sorry? It's an interesting question. What you're picking up on there, and it's a very interesting point, is that mixed inhibitors are binding somewhere else than the active site. And so in that sense, mixed inhibitors are allosteric inhibitors. But if we're looking at it kinetically, and we want to ask competitive, non-competitive, mixed, then that's the appropriate term. You're right. Mixed inhibitors, in fact, are allosteric. Good point. Oh, it's 20 after. Thank you all.
because I have people asking questions that are